audio is getting recorded. Their selective permeability allows them to serve as molecular gatekeepers. These membrane functions are accomplished primarily by a broad array of proteins and enzymes in and on membranes. At the cell surface, transporters move specific organic solutes and inorganic ions across the membrane. Receptors sense extracellular signals and trigger molecular changes in the cell. Ion channels mediate electrical signaling between cells, and adhesion molecules hold neighboring cells together. The other function of cell membrane includes the relative proportions of protein and lipid vary with the type of membrane, right? Reflecting the diversity of biological roles. For example, certain neurons have a myelin sheath, an extended plasma membrane that wraps around the cell many times and acts as a passive electrical insulator. The myelin sheath consists primarily of lipids, good insulators. In contrast, the plasma membrane of bacteria and membranes of mitochondria and chloroplasts, the sites of many enzyme catalyzed processes, contain more proteins than lipids in mass per total mass. This point basically sheds a very important light on a very important point, that though all biological membranes are made up of similar type of components, right, but still their composition varies depending upon the type of function they are involved in. If you can make this observation, that a myelin sheath whose primary function is to just transport signals, right, it is primarily made up of lipids, which are good insulators. On the other hand, membranes of cells, membranes of bacteria, or membranes of mitochondria and chloroplasts, which are highly active, which are highly functionally relevant, right? If you know mitochondria, there's a lot of things which goes on the membrane of mitochondria, like the ETS happens there, the ATPase component is completely present in the mitochondrial membrane. Similarly, for the chloroplast, the photosynthesis takes place. So all these organelles are highly functionally active, correct? And hence their composition, that is the amount of lipid, the amount of protein, they vary according to the function of a cell. The similar components will be present throughout the membrane, whether the membrane is from Golgi apparatus or lysosome or ER or mitochondria or chloroplast or a normal periphery or a normal plasma membrane of a whole cell. The components will remain same, but their composition will vary depending upon the functions in which those particular organelles and the membranes are involved in. Okay, this indicates that how dynamic our membrane is. This indicates that how dynamic our membrane is, right? So though we have similar composition components of a membrane like lipid, protein, cholesterol, sterols and carbohydrates, they will vary depending upon which membrane we are talking about. Whether we are talking about an organelle membrane, which is highly functionally charged. For example, if we talk about mitochondrial membrane, we know that there are a lot of enzymatic reactions takes place in mitochondria, and most of them are on the membrane, right? Similarly for the chloroplast. But on the other hand, if we talk about the membrane of a neuron, whose sole purpose is to transport a signal, so we might not need any accessory proteins in neuronal membrane, right? Essential proteins will be available, but they don't need any accessory proteins which might hinder in the function of that particular neuron, and the function is to just transfer a signal. Hence both neuron as well as mitochondria or chloroplast membrane have lipid and proteins associated with it. That is, the composition, the components of the membrane is same, but the composition, the amount of protein, the amount of lipid present in these membranes will vary. Okay, so within the cell, membranes organize cellular processes such as the synthesis of lipids and certain proteins, and the energy transduction that produce ATP in mitochondria and chloroplast. As I discussed earlier, like just now, that depending upon the function of an organelle, the composition of a membrane will vary. And also, as I've mentioned, we're going to discuss about different types of phospholipids, right? So the composition in that respect will also vary, that what kind of lipids are predominantly present in mitochondrial outer membrane, inner membrane. They vary, or they are different from the amount of or the type of lipid which are present in the nuclear membrane, or say ER membrane. Okay, so in all this manner, membranes are highly dynamic and very different structures depending upon their function. Or we can vice versa say that the function and the ability of a cell is determined by what kind of a membrane it is surrounded with. Okay, so proteins associated with membranes are essentially confined to two-dimensional space, where intermolecular collisions of membrane protein and lipids are far more probable than in the three-dimensional cytoplasm. So the efficiency of enzyme catalyzed processes organized within membrane can be vastly increased. So what this point is basically saying, that we have this sparsely distributed lipids and proteins, right? And I'm saying they are functionally relevant, they are functionally important. So for any protein, for example, if a ligand is coming, right, and if this is a membrane and this is a protein, so the ligand has to precisely sit here so that this protein can be activated and the cascade of biochemical reactions can take place. So for this ligand to sit exactly at this place, there is a very less probability if this protein is sparsely present throughout the membrane randomly, right? Hence, when we were talking about the membrane models, I said that though fluid mosaic model is the vastly accepted model of a membrane, lipid raft also came into action, right? So lipid rafts are basically these small domains which are formed transiently, which allow for an effective communication of a cell in between the cell actually. Okay, so these type of domains actually help in sending the signal and transmitting the signal very effectively without much loss in time. Correct? So if we talk about basic fluid mosaic model, right, as I've said, this is the vastly accepted model. So in fluid mosaic model, you can clearly see that this diagram is pretty similar to the one which we have seen earlier. So in this, as I have mentioned, that this is a lipid bilayer, right? So this lipid bilayer is majorly made up of phospholipids, and don't worry, we're gonna discuss their structure also maybe in the Tuesday class. Okay, today we're gonna focus more on the fluid mosaic model, and we're gonna see some of the like basic experiments how the fluid mosaic model was determined or was speculated. So when we see the fluid mosaic model, as I have mentioned, that this is a phospholipid bilayer. The head groups, these are the polar head groups, and they are hydrophilic in nature, while the tails, they are the acyl or fatty acyl side chains, and they are hydrophobic in nature. Okay, and you can see that there are different proteins which are embedded in the membrane itself. Correct? So there are different types of protein you can see. For example, if we look at this is the cytosolic side, this is the exterior side. So only on the exterior side we see modifications of the protein or lipid. As I mentioned, that these modifications were the addition of carbohydrates. Correct? So these are either glycolipids or glycoproteins depending upon where the carbohydrate modification has taken place. So there are different types of protein. Basic property of a fluid mosaic model of a membrane according to the fluid mosaic model is amphipathic structures. As I have mentioned, amphipathic means which has both hydrophilic and hydrophobic parts, and hence our lipid bilayer is of amphipathic nature, right? And different types of protein are present in the membrane. For example, one type of protein which is associated with the membrane is called peripheral membrane proteins. So peripheral membrane proteins are the ones which are only associated with the one leaflet of a membrane, right? They are not inside the membrane, but they are associated at the surface of a membrane. So these are called peripheral type of membrane proteins. Now these peripheral type of membrane proteins can either be directly associated with the lipid membrane, or they can be associated with certain linkers. If you remember, we in the previous class we talked about GPI anchored proteins, right? So they are associated with the membrane but only to a single leaflet of a membrane. Another type of protein is called the integral membrane protein. So as you can see, integral membrane proteins are the ones which are transversing the membrane completely. That is, they're transversing the lipid bilayer completely, and a major chunk of the protein is in between the lipid bilayer. Okay. The third type of protein is called a lipid anchored proteins, correct? So these are the like GPI anchored one which I was talking about. So it is a type of peripheral protein as it is associated with the periphery of the membrane, but still they will be associated with certain anchors which will dig deep into the membrane and associate the protein with it, correct? And because of this nature, that there will be certain type of proteins which will be present on upper leaflet, there are certain types of protein which will be present at the lower leaflet, certain proteins which are completely transversing the membrane bilayer, and also, as I've mentioned, different types of lipids will be present in upper leaflet and lower leaflet. All these are the source of asymmetry in the membrane. So if someone asks you what is the fluid mosaic model, so you can clearly say that these are the most commonly accepted model of a cell membrane. In this, the protein icebergs float in the sea of lipid. Okay, as you can see that these are the lipid, the sea of a lipid, and these proteins are floating in here, and these are different types of protein. And the source of asymmetry, where the asymmetry comes into picture, the asymmetry comes into picture for a lipid via both from the type of lipid as well as the proteins which are present inside the membrane, correct? So to understand more deeply into this, I have this particular video which very beautifully explains the fluid mosaic model. Let me share my screen with you guys. I hope you can see and hear it, right?
Let's explore the fluid mosaic model of cell membranes. Now, why is it called the fluid mosaic model? Well, if we were to look at a cell membrane, and just to be clear what we're looking at, if this is a cell right over here, if this is a cell, and this is its membrane, this is the membrane. It's kind of what keeps the cell, the inside of the cell, separated from whatever is outside the cell. We're looking at a cross-section of its surface. We're looking at a cross-section of its surface. We're down here, this is inside the cell. If we look at it relative to this diagram, this is inside the cell, and this is outside. This is outside. And when you zoom in, this little part right over here, this is actually a phospholipid bilayer that forms it. And so when you hear that, you might say, well, what is a phospholipid? And that's a good question, because when you understand what a phospholipid is, it starts to make sense why it would form a bilayer like this and why it's the basis for so many membranes in biological systems. So this is indicative of a phospholipid. And as its name implies, and let me write that down, this is a phospholipid. It's a lipid that involves a phosphate group. And in general, the word lipid, and we have a whole video on lipids, means something that doesn't dissolve so well in water. And that's true as the case of this phospholipid. You have these hydrocarbon tails that are coming from fatty acids. And so these hydrocarbon tails, they have no obvious charge or no obvious polarity. We know that water is a polar molecule. That's what gives us its hydrogen bonds, and it's attracted to itself. But these don't have those, and so they're not going to be attracted to the water. The water's not going to be attracted to them, and so these tails are hydrophobic. So you have hydrophobic tails, And these are really kind of the lipid part of the phospholipids. And then you have the phosphate head. You have the phosphate head right over here. And as you can clearly see, this has some charge. Charged molecules do well in polar substances like water. They're going to dissolve well. And so this part right over here is going to be hydrophilic. Hydrophilic. And actually, molecules that have a hydrophilic part and a hydrophobic part, there's a special word for them, amphipathic, a word that I sometimes have trouble saying. So phospholipids are amphipathic, which means that they have both a hydrophilic end, a part that is attracted to water, and a hydrophobic end that is not attracted to water. And hopefully that starts to explain why they organize themselves in this way. Because you can imagine the hydrophilic heads are going to want to be where the water is, which is going to be either outside the cell or inside the cells. And the tails are hydrophobic. The water is going to go away from them, or they're going to go away from the water. And so they're just going to face each other, and they're going to be on the inside of the membrane. But the really cool thing is a structure like this, having this amphipathic molecule, allows things like these lipid bilayers, I should say, to form. And it's actually fascinating if we think that if you go far back enough, even before life in cellular form formed, that you might have had phospholipids spontaneously forming these spheres of that where you have a bilayer, a lipid bilayer. So you could imagine something. Let me see if I drew a cross section. Let me see if I could draw it relatively neatly. So I think I'll draw half of it just because I'll draw the whole thing. Hopefully you get the idea. So that would be one layer of the phosphate heads facing the outside. This is the inner layer. And I'm doing a cross section right over here. And then you have your hydrophobic tails. So your hydrophobic tails, let me do that in a different color. So your hydrophobic tails, I think you get the picture. We have a bunch of hydrophobic tails on either end. And then you could spontaneously form a structure like this, which starts to feel like, hey, well, maybe there's a protocell forming. And obviously to actually have real life, you have to have some form of information that can be passed on. And you have to have some type of metabolism. And the cell is living in all of the definitions of life. But at least this basic structure of the cellular membrane, you can imagine how it forms in kind of in a pre-life state even, by virtue of amphipathic molecules like a phospholipid. So fair enough, we're able to form this phospholipid bilayer. But what are all these other things that I have drawn here? Well, these are proteins, and these are examples of, so this is a protein right over here, this is a protein, this is a protein, and I just drew some kind of blobs to be indicative of the variety of proteins. But the important thing to realize is when you think of cells, there's all of this diversity, there's all of this complexity that is on or embedded inside of its membrane. So instead of just thinking of it just kind of as a uniform phospholipid bilayer, there's all sorts of stuff. Maybe if we view this as a cross-section, there's all sorts of stuff embedded in it. And we see it right over here in this diagram. You could say there's a mosaic of things embedded in it. A mosaic is a picture made up of a bunch of different components of all different colors, and you can see that you have all different components here, different types of proteins. You have proteins like this that go across the membrane. We call these transmembrane proteins. They're a special class of integral proteins. You have integral proteins like this that might only interact with one part of the bilayer, while these kind of go across it. You have things like glycolipids. So this right over here, this is a glycolipid. Glycolipid, which is fascinating. It lodges itself in the membrane because it has this lipid end. So that's going to be hydrophobic. It's going to get along with all of the other hydrophobic things. But then it has an end that's really a chain of sugars. And that part is going to be hydrophilic. It's going to sit outside of the cell. And these chains of sugars, these are actually key for cell recognition. Your immune system uses these to differentiate between which cells are the ones that are actually from my body, the ones that I don't want to mess with, the ones I want to protect, and which cells are the ones that are foreign, the ones that I might want to attack. When people talk about blood type, they're talking about, well, what type of specific glycolipids do you have on certain on cells? And there's all sorts of, and that's not all we're talking about when we talk about glycolipids as kind of a way for cells to be recognized or to be tagged in different ways. So it's a fascinating thing that these chains of sugars can lead to such complex behavior and frankly such useful behavior from our point of view. But you don't just have sugar chains on lipids, you also have sugar chains on proteins. This right over here is an example of a glycoprotein. Glycoprotein. And as you can see, when you put all this stuff together, you get a mosaic. And I'm actually not even done there. You have things like cholesterol embedded. Cholesterol is a lipid, so it's going to sit in the hydrophobic part of the membrane, and that actually helps with the fluidity of the membrane, making sure it's not too fluid or not too stiff. So this is cholesterol right over there. So you see this mosaic of stuff, but what about the fluid part? And I just talked about cholesterol's value and making sure that it's just the right amount of fluidity. What's neat about this isn't a rigid structure. If this thing were to be jostled around a little bit, or maybe it would be plucked out somehow, the phospholipids would just spontaneously rearrange to fill in the gap. You can imagine these things are all flowing around, that this membrane actually has a consistency of kind of oil or salad dressing. So it isn't this it isn't like a rubbery texture like you might imagine a membrane, like a balloon. It's actually fluid. These things can move around, but even though it's fluid, it's good enough to separate the two environments, the environment inside the cell from the environment outside of the cell. And that's where the name fluid mosaic model comes from.
Let me share my screen. I hope you can see my screen, my presentation. Yeah, I guess it might be visible to you now. Yeah, so I hope that you now have a very, very, very basic idea about the components of a cell membrane, as well as why we are calling it fluid mosaic model, right? So this fluid mosaic model has two words. The first one is the fluidity, the fluid part of the membrane, and the mosaic part of the membrane. So mosaic, I guess most of you guys know what it means. Mosaic is basically the random distribution of things, right? And fluid is obviously the ability to flow. So our lipid membrane is first fluidic in nature, and this fluidity comes because of the nature of lipids which are present, and there as it's a lipid, right? It's a organic molecule, so it will have certain freezing temperature, melting temperature, all the chemical properties of a lipid, right? And then why it is called mosaic? Because these proteins are randomly distributed inside the membrane. They can be either on one leaflet, either the upper leaflet or the lower leaflet. These can have certain modifications which will be called glycoproteins, or they can be completely traversing the membrane. Though it is called mosaic in nature, but this is my personal opinion that this random distribution of proteins in the membrane is not that random, right? So as we are talking about lipid rafts, so the whole sole purpose of lipid raft is to make these proteins more accessible to the outside world, whatever the function is, irrespective of whether it's a transporter or a receptor or anything, right? So if you make it localized, the probability of the ligand or a small molecule in actually hitting the protein increases, right? So though the distribution is random, but still it is not completely random, and we do not understand, or at least I am not aware of such literature which actually suggests that the distribution is not random. it will be random in the majority of the sense, but still I feel like the cell membrane and the cell is too smart to do anything randomly. Okay, so now if we see some of the characteristics of the fluid mosaic model. So what exactly is a fluid mosaic model, right? So a fluid mosaic model explains various characteristics regarding the structure of functional cell membrane. According to this biological model, there is a lipid bilayer. I hope that these words are now completely engraved in your mind now. Lipid bilayer, two molecules thick layer consisting primarily of amphipathic phospholipids, in which protein molecules are embedded. The phospholipid bilayer gives fluidity and elasticity to the membrane. Okay, small amounts of carbohydrates are also found in the cell membrane. The biological model, which was devised by Singer and Nicholson in 1972, describes the cell membrane as a two-dimensional liquid that restricts the lateral diffusion of membrane components. Such domains are defined by the existence of region within the membrane with special lipid and protein cocoon that promote the formation of lipid rafts of proteins and glycoprotein complexes. Okay, so it's a very simplistic way of defining a fluid mosaic model. I really hope that it is clear to you that what are the components of a membrane, what are the major functions of a biological membrane, what are the properties of a biological membrane. So from next class onwards, we're gonna discuss some physical parameters, because these all are physical structures, some physical parameters on which we can describe our membrane, right? But before that, you guys must have one question in mind, right? Though we are saying that fluid mosaic model is the most widely accepted model for describing a cell membrane, but how come we have come to this conclusion that a cell membrane or a biological membrane is fluidic in nature, first part of the model, and the distribution of proteins, or it's a mosaic distribution of components. So how come we are actually understanding these things? So to get to this model, there are n number of experiments into play, but there are certain defining experiments which actually put a nail on the concept that a membrane is fluidic in nature and the distribution of proteins and the component inside the membrane is of random distribution and hence mosaic in nature. So first we'll see how or what were the experiments which actually said that the membrane is fluidic in nature. So before understanding that, these are the basic components of a cell membrane. I hope that I'm repeating it so many times that now you guys might be saying this even in your sleep, right? So there are four major components of a cell membrane. Phospholipid, which make up the majority of the membrane, and they are making the lipid bilayer nature or the membrane itself. Carbohydrates, they are the ones which are present only on the outer side of the membrane. They can be either linked to a protein or they can either link to the lipid which are present, correct? Then the cholesterol or the sterols, and last but not the least, the proteins. So the location is pretty specific and pretty common, that the membrane is asymmetric in nature, right? So their distribution will also be asymmetric. Phospholipid, as it is making the plasma membrane, hence the location will be like this is the plasma membrane. Okay, carbohydrates, as you have seen in the video as well as in the representative structures of a membrane, the carbohydrates are only present on the outside of a membrane and they help in providing the identity to the cell itself. For example, your blood group, your type of cell, whether it is CD4 type of cell, CD8 type of cell, all these CD, when we say the markers of a cell, these markers are at the end of the day are the carbohydrates or the proteins which are associated with the outside of a cell. Then cholesterols are present in between the lipid bilayers, as we have seen in the representative structure of a lipid bilayer. Then proteins, so there are different types of proteins, correct? They can be either present on one leaflet of the protein, they can be attached to the lipid bilayer via anchors, or they can completely transverse the membrane being the integral membrane protein, and correct. And even in the integral membrane protein, we're gonna discuss that there will be a multi-pass protein, single pass protein, right? So there are different types of integral types of protein also, correct. Don't worry if you are getting confused, we gonna like see everything in a tabular form and it will be easier for you guys. Then the function of these components. So phospholipid, it provides selective permeability to the cell membrane. I hope that this is clear to you guys, right? As we have discussed, it is amphipathic in excuse me. Can you please unmute yourself, Shreyash? Sorry, mute yourself. I'm sorry, you are already unmuted. Yeah. So phospholipids, they basically provide selective permeability to the cell. So why we are saying this? If you remember, when we were talking about this structure, we said that it has a polar head group and a hydrophobic tail, right? So it will only allow small molecules to pass through them, and for other transportation of Shreyash, if you have any question, please unmute yourself, otherwise keep yourself muted. Yeah, so it provides selective permeability to the cell membrane. The hydrophilic phosphate side is outwards and hydrophobic inwards. Yeah, so I have muted you. Yeah, so these plasma membrane allow selective transportation of molecules. They will allow small polar molecules to pass through them, but they won't allow bigger molecules to pass through them, and for that we need proteins, right? And carbohydrates, it helps in cell to cell recognition and also provide the marker to the cell, act as a marker. Then cholesterol, it helps the plasma membrane to retain its fluidity. Then proteins, obviously they are very diverse, and describing them in one sentence is not a justice to them, but later part when we will be done with our basic structure and basic components of a cell membrane, we're gonna solely focus on the protein at the end of the day, right? So proteins, these form channels to allow the movement of molecules. So as I was mentioning, what were the experimental evidences that actually lead to the conclusion that the membrane is fluidic in nature, comes from a labeling experiment and X-ray diffraction techniques. So basically the fluid property of functional biological membrane had been determined through labeling experiments and X-ray diffraction. These all are visualization and you can visualize components moving if the property we are determining is fluidic in nature, right? So one way to determine whether the component or the biological section which we have with us is fluidic is it will be easier to see if something is moving, right? If something is moving, we can say that it is not rigid and it is fluidic in nature. So this is exactly what the scientists have done to understand the fluidic nature of the membrane. So an important experiment that provided evidence supporting fluid and dynamic biological was performed by Fry and Edidin. They used Sandai virus to force human and mouse cells to fuse and form a heterokaryon. Using antibody staining, they were able to show that the mouse and the human proteins remained segregated to separate halves of the heterokaryon a short time after cell fusion, right? So now if you have basic idea about how these experiments are done, so you take one mouse cell and you can take one human cell and you just fuse them with the help of virus, correct? Because virus can transfect and they can fuse. So after a while, and they are obviously labeled differently. For example, a human cell will be labeled red in color and a mouse cell will be labeled green in color. So if they are separate and they are not fluidic in nature, so even after some time what will happen, you can easily see two halves. One half will be red, saying that this is human part of the cell, and one part will be green, indicating that this is the mouse part of the cell. But on the other hand, if the membrane is fluidic in nature, right, so what will happen after some time? They will mix among themselves, right? So this is exactly what has happened. The protein eventually diffused, and over time the border between the two halves was lost. Lowering the temperature slowed the rate of this diffusion by causing the membrane phospholipid to transition from a fluid to a gel phase. Singer and Nicholson rationalized the results of these experiments using fluid mosaic model, right? So after some time, what happened is that both red and green dye were mixed when the scientists observed this hybrid heterokaryon, indicating that proteins are actually randomly moving. So proteins can only move when they are present in fluid, right? If they are present on a solid surface, they won't be able to move, and they will stay at the place wherever the labeling was done, right? So this particular type of experiment basically indicated that membranes are in fluidic in nature, right? Also, this is the article which I have given the reference here. This tells you another basic technique which was used to understand the mosaic nature of the membrane, and this technique is called freeze fracture electron microscopy. So freeze fracture electron microscopy, basically what it does is you freeze the membrane in whatever state it is, then you slice the membrane parallel to the leaflet, like in from between the two in between the leaflet, and you just observe it. How are the things there? Are they randomly distributed, or is there any particular order associated with it? So for this, I was not able to find much for visual representation purposes, but still I was able to get something out of it. So this is a very brief description of freeze fracture electron microscopy.
But as it says now, look at that structure. About the fluid mosaic model, used to be called the fluid mosaic theory of membrane structure. You can take purified membranes or even whole cells, freeze them in ice, go get a hammer and chisel and tap at the frozen ice block containing the membranes, and what will happen is the membranes will fracture along the line of least resistance, the interaction of the hydrophobic fatty acid tails of the phospholipids. So you will split the phospholipid bilayer into two halves. And there's a cartoon that I drew for you. And the idea is that if you split the membrane in half, some of the proteins will travel or stick with one half of the phospholipid bilayer, some with the other. I've drawn it so that they're all on one side, which is typically what you see when you actually look in the electron microscope. And what you can see is if there's a protein stuck to one side of the phospholipid bilayer, because it was frozen, nothing had a chance to shift, and you can actually see a groove or a hole even where that same protein was embedded in the other side of the phospholipid bilayer. This kind of electron microscopy tells us that there are indeed proteins embedded in the phospholipid bilayer, that they are not, for example, sitting on either side of the membrane that is interacting, say, just with the phosphate heads of the phospholipids, but they're actually embedded and they stick all the way through a membrane. So all the ones shown here, with one exception, called a peripheral membrane protein, are called transmembrane or integral membrane proteins because they span the entire phospholipid bilayer. The illustration shows the oligosaccharides, and so without my telling you, you should be able to identify what's outside and what's inside relative to this bit of plasma membrane. The glycoside-linked sugars in the glycopeptides, and in one case a glycolipid, must be on the outside of the cell.
Right, so this is the very basic way how Singer-Nicholson combined these type of experiments to come up with a model which is called fluid mosaic model. And the article which I was telling you about, that I have given the reference, is this, like freeze fracture electron microscopy. So this is not an old, so-called old technique, but yeah, it's a very useful technique, especially if you want to understand the distribution of things specifically for a biological sample. So I'll just go through the abstract so you guys have a little bit idea about this type of technique. The freeze fracture technique consists of physically breaking apart, fracturing a frozen biological sample. Structural detail exposed by the fracture plane is then visualized by vacuum deposition of platinum carbon to make a replica for examination in the transmission electron microscope. The 4 key steps in making a freeze fracture replica are rapid freezing, fracturing, replication, and replica cleaning. In routine protocol, a pre-treatment step is carried out before freezing, typically comprising fixation in glutaraldehyde followed by cryoprotection with glycerol. An optional etching step involving vacuum sublimation of ice may be carried out after fracturing. Freeze fracture is unique among electron microscope technique in providing planar views of internal organization of membranes. Deep etching of ultra rapidly frozen sample permits visualization of the surface structure of cells and their components. Images provided by freeze fracture and related techniques have profoundly shaped our understanding of the functional morphology of the cell. Okay, so I hope that you guys can go through that article, and I will upload this slide as soon as the portal is ready along with all the videos of this particular session. If you guys have any question, feel free to either ask here, or you have my email ID, you can drop me queries anytime. And I hope to see you guys in the next session, which will take place on Tuesday. Okay, bye. Thank you for your time.